I'm Roxanne Barrion from CJ Bio APAC Technical Solutions Center, and today I will be discussing about low crude protein diets in broilers and some recent updates. I'll start my presentation with a short introduction which led to dietary CP reduction and its advantages in animal production, followed by the recent updates on low dietary CP reduction and the conclusion of this presentation.
The focus of most countries is to be able to establish food security and sustainability for the ever-growing population. Two of these sustainable development goals that CJ has been focusing is zero hunger and partnership for the goals. For us to achieve this, we aim to reduce the dependency to soybean meal for animal feeding and instead increase its availability for human consumption or be able to use the land to grow edible grains. With a reduction in the inclusion of soybean meal in animals' diets, it may also reduce the environmental nitrogen pollution and carbon dioxide equivalents. On this regard, feed security of the general population could be achieved while also promoting an antibiotic-free meat and maintaining animal welfare. When formulating diets, nutritionists tend to consider several factors that somehow prevents them in reducing CP, such as the published nutrient recommendation of the breed, age, stage of production, such as if the animals are reared for meat, eggs, milk, and others, health status, changes in climatic condition, management, and housing, or some other factors that may influence the requirements of the animals, especially for protein or shall we say amino acids. Unlike with energy, there are several kinds or types of grains and other alternative raw materials that can fulfill the animal's energy requirement. Whereas for protein, there are only limited numbers of feedstuffs that can provide this nutrient and is normally limited to soybean meal, animal proteins, fermentation biomasses, or single-cell proteins. And based on most practitioners' experiences, cost, quality, quantity, and magnitude of anti-nutritional factors are some of the restrictions that we can consider when formulating diets with these ingredients. This is because only few countries produce soybean meal, which is insufficient most of the time since not only animals consume it, to meet the ever-growing demand for this commodity together with its rising prices. As a result, escalating prices of animal feeds is inevitable. Feed producers already tried several strategies to ameliorate the rising feed cost by embracing alternative feed ingredients, optimizing or maximizing the matrix values or nutrient contribution of the enzymes or emulsifiers, eliminating feed additives with no nutrient profile, or feed cost savings claims. Then adjustment on nutrient specifications, and finally, the thing that we don't want to happen is the last option, which is to reduce flock population to lessen possible losses. With low CP diets, lowering feed cost is possible. And eventually, the animal production cost, as shown on this formulation simulation, where a 1.8% CP reduction resulted to around 3 USD or $3 savings per metric ton, or around 3,000 USD savings per month if the feed mill is producing 1,000 metric tons per month, which can be used for other operational costs. The word crude in crude protein refers to the fact that not all dietary nitrogen is fully utilized by the body for protein deposition. Only 45% of the total dietary nitrogen is used and retained by the body for growth and other metabolic functions, whereas the remaining 55% is excreted in the form of ammoniacal nitrogen and nitrogen in the soil. To reduce the 55% dietary nitrogen excretion by the body to the environment, dietary CP level is recommended to be lowered, leading to a decline on the inclusion rates of protein-rich ingredients, particularly soybean meal. Soybean meal remains to be the most important and preferred source of high-quality vegetable protein for animal feed production. A 1% reduction on the total dietary CP may lead to a decrease on the inclusion rates of soybean meal by around 3 to 4%. However, with the reduction on the levels of soybean meal on the animals' diets, the amount of essential and non-essential amino acids will be reduced significantly and may allow increased incorporation rates of crystalline or supplemental amino acids in the diet. Additionally, around 9 to 15 kilograms of carbon dioxide equivalents is reduced for every kilogram of soybean meal that will be deducted on broiler feed formulation. The question is, how shall we reduce crude protein? Based on our own researches and findings, to be able to successfully reduce dietary CP levels is to first establish the SID lysine level of the formulation based on the requirements of the breed and use the appropriate ideal amino acid ratios of all the 10 essential amino acids relative to lysine. There are cases that breeds will require some non-essential amino acids when considering the essential amino acids such as cysteine with methionine and tyrosine with phenylalanine. After fulfilling these nutrient requirements, we can then eliminate the restrictions in CP values. However, there will be cases when the total ratio of non-essential amino acid relative to essential amino acids must be taken into consideration as we further reduce the dietary CP contents to prevent unwanted responses from the animals. Moving on with the advantages of low CP diets, we will start with the positive effects of feeding low CP diets or CP levels that lessen the incidence of enteric diseases in animals caused by common and several pathogenic microorganisms such as Clostridium perfringens, Eimeria, and E. coli. This can be explained by the fact that high-protein diets have higher acid binding capacity and increased pH of the gut, of the gastrointestinal tract to nearly neutral conditions, which provide a favorable environment for the proliferation of pathogenic bacteria such as Bacteroides and Clostridium species, thus reducing the amount of protein reaching the large intestine by selecting highly digestible protein ingredients or lowering the dietary protein level may help to alleviate wet litter in poultry or post-weaning diarrhea in swine. This diagram from the paper of De Cesario and colleagues illustrates the population of lactobacilli were significantly higher in the Zika of broilers fed with low CP diets, which indicates that low CP diets may hinder the growth of pathogenic microorganisms that may lead to infection. Excreted nitrogen and moisture and uric acid were the parameters that were observed by Namrod and colleagues as dietary CP is reduced with essential amino acid supplementation. Similarly, the results were observed from the data showed by Ospina Rojas indicates a statistically significant reduction in liter nitrogen, ammonia emission, and liter pH with a CP reduction of 3% but with essential amino acid supplementation. And no changes in FCR were observed. This indicates that the rate of protein accretion is not dependent on dietary CP levels, especially if the amino acid requirements of the animals are met. Additionally, the incidence of foot pad dermatitis in broilers can be reduced as we lower the CP levels. And in some countries, chicken feet has economic value. The reason for this can be attributed to the less water intake of the animals as we lower the CP levels. We need to remember that excreting excess nitrogen from the body requires a lot of water, which of course will be eventually excreted as well as from the body. An increase in litter moisture softens the surface of the chicken feet and invites pathogenic microorganisms to proliferate, causing foot pad dermatitis. Moving on with the recent updates on low CP diets in broilers, despite the number of papers showing the positive effects of low CP diets, there were still some journals or experiments that demonstrate its negative impacts in broilers. There are several factors identified that could be the reason for such responses. One of these is protein synthesis demands energy as an input of 5.35 kilojoules or around 1.28 kilocalories that is required for the synthesis of 1 gram of protein. Protein efficiency is greatly influenced by the harmonious provision or availability of amino acids and non-amino energy sources, essentially from glucose or starch, to sites of protein synthesis. Relying on intact protein sources may delay the immediate availability of the amino acids for protein accretion because of the rapid digestion of starch as energy source. Dietary energy is used for protein synthesis and also for excreting excess nitrogen from crude protein. Deaminating or excreting excess ammonia requires 4 ATPs for each molecule of urea, and each ATP is equivalent to around 85 kilojoule or metabolizable energy of around 20.32 calories. This is estimated to be around 10.2% of the energy in protein. Hence, as dietary CP rise, the heat increment and urinary energy also increases, and the efficiency of energy for production purposes decreases. This is one of the reasons why most researchers are looking into the application of low CP during heat stress conditions. Lowering dietary crude protein do not only contributes to the heat stress alleviation in poultry, but also allows the other amino acids to contribute its functional roles in the animals. Such amino acids are arginine, the branching amino acids leucine, valine, and isoleucine, phenylalanine, tyrosine, glycine, and glutamine that plays a significant and functional role in thermoregulation, especially using their crystalline forms, which are highly digestible and immediately enter the blood for absorption with low requirement for dietary energy compared to intact proteins. Breast muscle yield is a good measurement to determine the amino acids bioavailability in poultry diets. And based from this study, when lowering further the dietary crude protein levels beyond 3%, increasing the SID lysine to ME ratio is the best approach to achieve the same performance of the broilers fed with standard CP levels. Lowering dietary CP levels also allows the removal of excess amino acids, which most of the time causes an imbalance. These amino acid imbalances give rise to a surplus of amino acids, which are later catabolized and increase the possibility of excessive levels of ammonia being generated. From this deletion study, wherein we try to identify the orders of limiting amino acids on broilers fed with low crude protein diets, and interestingly, lysine and methionine were found to be co-limiting in the first position, followed by threonine, then valine, and then arginine and tryptophan. During the time of the study, histidine was not yet commercially available, which may have affected the positioning of isoleucine. Another interesting fact from the study of Shao and colleagues suggests that lowering dietary CP do not negatively influence the growth and development of immune organs, which translates to unaffected immune response of the birds. Furthermore, the plumage quality, breast, skin, and incidence of blisters and heartburn and swelling of broilers fed with low CP diets were not found to be different from chicken fed with standard CP levels. Oppositely, the incidence of foot pad dermatitis were shown to be less as we lower the dietary CP content. Another interesting information that I would like to share is the impact of low CP in meat quality. From this experiment, it resulted in an increase in ultimate pH of breast meat, and according to several authors, the ultimate pH is negatively correlated to the glycogen content of the muscle. We assumed that the provision of a more balanced amino acid profile associated with the reduction in CP content reduced the excessive amino acids, decreasing the amount of nutrients available for glycogen production or storage through amino acid catabolism. Several studies have indeed shown that the ultimate pH responds to dietary treatments and in particular to variation in the lysine, methionine, or protein. Moreover, it was shown that the increase in ultimate pH or breast meat was associated with a lower lightness and trip loss. Finally, with our conclusion, animals are not growing on crude protein provided that all amino acid requirements are met. Reducing crude protein lowers nitrogen excretion and lessens environmental pollutions, and thereby promoting sustainability of the animal industry. Dietary CP reduction helps to decrease the prevalence of enteric diseases and thereby promoting animal welfare and reduces the usage of in-feed antibiotics and antibiotic usage in animals. CP reduction cuts feed costs and eventually farm production or farm or animal production costs, and thereby increasing the return of investment. Crystalline amino acids are required in low CP diets to ensure that the amino acid supply covers the requirement of the animals. Excess dietary protein can increase heat increment in animals, and ideal amino acid can be easily balanced by crystalline amino acids which are rapidly absorbed and metabolized than intact proteins. Dietary CP reduction poses more benefits to the society, economy, and environment. With this, thank you for listening, and we will be very happy to answer any additional information that you may need through this email. Again, thank you so much and enjoy your day.